Good morning, students. I think everyone is fine. And last day we have discussed many points related with the structure of cell organelles. What are called cell organelles? They are the different components in the cell. Mainly we have discussed the endoplasmic reticulum, Golgi apparatus, then lysosomes, all these things, and mitochondria. All these cell organelles we have discussed the last class. Then we will see what are the different points we discussed in the last class. It was about endoplasmic reticulum. There are two types of endoplasmic reticulum. What are they? Rough endoplasmic reticulum and smooth endoplasmic reticulum. And what is the function of rough endoplasmic reticulum? Rough endoplasmic reticulum is the site of protein synthesis. Then it produces proteins. Then we discussed the smooth endoplasmic reticulum is the site of synthesis of what? Lipids and fats. Okay, all these proteins, lipids and fats are used for the construction of cell membrane. That process is known as membrane biogenesis. Okay, so here in the endoplasmic reticulum, proteins are made with the help of ribosomes. Where are they present? They are present. Ribosomes are present on the surface of the rough endoplasmic reticulum. So, this rough endoplasmic reticulum, with the help of ribosomes, they can produce proteins. And smooth endoplasmic reticulum, they can produce fats and lipids. These all are required for the construction of what? Membrane. Which membrane? Cell membrane. So, this process is known as membrane biogenesis. Then next we discussed about the Golgi apparatus. Then they are discovered by one scientist called Camillo Golgi. Then it consists of a system of membrane bound vesicles, and this Golgi apparatus carries the materials from endoplasmic reticulum to different sites in the cell, in different targets in the cell. So here proteins are made in endoplasmic reticulum. Fats are made in the endoplasmic reticulum, or lipids are made in the endoplasmic reticulum. All these materials that are produced in the endoplasmic reticulum are stored, modified and transported to different sites or different parts of the cell. That is the function of Golgi apparatus, and it has an important role for what? In the formation of lysosome. Then we discussed about lysosomes. Lysosomes are known as the suicidal bags of the cell. They are also like vesicles. Why are they called the suicidal bags of the cell? Because if something, or if any cell organelle is damaged, or the cell itself damaged, then what may happen? The lysosomes present in those cells will completely destroy all the cell organelles in the cell. So then they are called the suicidal bags of the cell. So it's a site of waste disposal in the cell. All the unwanted foreign materials are deposited in this particular site, and this site is known as what? Lysosomes. Okay. After that we discussed the mitochondria. Mitochondria is known as the powerhouse of the cell. Why? Because it provides energy for what? For performing many activities, chemical reactions in the cell. And this energy is stored in the form of what? ATP. What is ATP? Adenosine triphosphate. And this ATP is also known as what? The energy currency of the cell. Energy currency of the cell. And the stored energy is utilized for many chemical activities in the cell or many other functions. Okay. And mitochondria has two coverings, an outer covering and inner covering. Outer covering is inner covering is very deeply folded, and so that many ribosomes are or many ATP molecules are present inside. And mitochondria, they have their own DNA, and they have their own proteins. So as they have proteins, they do not depend on any other cell organelle for proteins. Okay, so they can prepare their own proteins with the help of ribosomes present in them. Okay. Now today we are going to discuss about plastids, the next cell organelle, plastids. Plastids, plastids. Okay. Plastids are present only in plant cell. It means that in animal cell, plastids are absent. Plastids are absent in animal cells, only in plant cells, or in plants we can find plastids. Then there are two types of plastids, leucoplast and chromoplast. What are they? Leucoplast and chromoplast. Leucoplast means they are colorless, means they do not give any color to the cell, just they have some protein, granules, starch or oil. Leucoplasts. They are colorless plastids. Colorless plastids, and they store, leucoplasts store some granules, some protein granules, some oil or starch, etc. These materials are stored in leucoplast. Okay. But some other plastids are there which are having chlorophyll in them. Such plastids are known as what? Chlorophyll. Means in some plastids it contains chlorophyll pigment. You know what is chlorophyll? Green colored pigment. This green colored pigment called chlorophyll is present in some of the plastids. So such plastids are called chloroplast. Chlorophyll is present, so that chloroplast. Leucoplast means colorless, no color, no pigment present in them. But this leucoplast stores what? Starch, oil, then protein granules, etc. Okay. Next, chromoplast. Chromoplasts are the plastids that provides what? The color to the plants. So this chromoplast is present in different parts of the plant. It may be present in the root, or in the stem, or in the leaves, or in the flowers. You know, pigment, color pigment, different colored leaves we can see, different colored stems we can see in some plants. In some plants the color of the fruit is different colored, very attractive colored fruits, flowers, leaves are there in plants. All those colored pigments they get from, or plants get from what? From this chromoplast, or because of the presence of this chromoplast only, those all parts get what? The different attractive colors. Okay. So chromoplasts are colored plastids and gives color to the plant, and it has same this plastids, they have a similar structure like mitochondria, and they also have their own DNA and proteins in them. So they also do not depend on any other organelles for proteins. Okay, that is about plastids. Once again, plastids are present only in plant cell. Second one, there are two types of plastids. What are they? Leucoplast and chromoplast. Some of the plastids they may have a green pigment called chlorophyll. Such plastids are called chloroplast. Okay. Now leucoplast. Leucoplasts are colorless plastids, and these plastids contain what? Or they store some materials like protein granules, oil, starch, etc. In chromoplast, they are colored plastids. They provide color to the plants, to the leaves, to the fruits and flowers. Okay. And here, this chromoplasts, they are giving color to them. So attractive color to the plants. And plastids they have a similar structure like a mitochondria. Before we discussed what are called mitochondria. Okay. So, and they have their own protein, and they have their own RNA, because they have ribosomes, so that they can produce their own protein. Next, vacuoles. Vacuoles, they are present in both animal cell and plant cells. But in plant cell, we can see they are large in size. Vacuole is large in size, where in plant cell, but in animal cell, these vacuoles are very small. So, about in plant cell, about 50 to 90% of the volume of the cell is occupied by vacuole. Suppose if we consider a cell, a plant cell, we can see that the maximum volume, maximum space in the cell is occupied by vacuoles, where in plant cell. But in animal cell, these vacuoles are very small. And these vacuoles they are filled with cell sap, and so that the cell sap is present, means completely filled like a bag, and it provides very turgidity and rigidity to the cell. Turgidity and rigidity means, we know that suppose if we blow a balloon, blow air into the balloon, then what happens? We can see that the balloon becomes very large in size. So we may feel that it is going to break. Such a feeling, a turgidity, inside there is a pressure, we may feel that it is going to break up. In such a way it is going to burst. So in such a way we feel. So it provides that such a feeling. What is it called? Turgidity or rigidity, strength and turgidity to the cell. That is a function here. So cell sap is present in vacuoles, and this cell sap is filled maximum in this vacuoles. So it is having a property, or it is having a structure like a having rigidity or turgidity. Okay. Now these vacuoles contain, what are the contents in the cell? What are the components in vacuoles? These vacuoles contain amino acids, sugars, and some other various organic acids, then proteins. These are the contents. Many sugars, then amino acids, then various organic acids and proteins. All these are present inside this cell sap or in the vacuole. Okay. So we know, again I say, vacuoles, they are present in both the cell, but in plant cell they are large in size, very big in size, and in animal cell they are very small. Then in this plant cell, about 50 to 90% of the total space is occupied by vacuole, but in animal cell they are very small in size. And what is inside? Inside this vacuole there is cell sap. Cell sap is filled inside. So we feel, or it has turgidity or rigidity. Okay. Next, inside the cell sap or in the vacuoles there are some materials. What are the components present inside? Amino acids, sugars, then various organic acids and proteins. These are the contents. In unicellular organisms, before we discussed, like amoeba, paramecium, they have what? Food vacuole. And this food vacuole helps to expel the excess water and waste from them. So once they absorb water inside along with the food, so this water is expelled out, or it will be released out from this cell with the help of what? This vacuoles. That is the use of vacuole in unicellular organisms. Okay, these are the main points described in this chapter. Okay, we have discussed many things. What is cell? Who discovered cell? What is cell theory? Who proposed cell theory? Then we discussed multicellular organisms, unicellular organisms, then eukaryotic cells, prokaryotic cells. Then we discussed, after that we discussed about cell wall, nucleus, cell membrane, then cytoplasm, and we discussed different cell organs like endoplasmic reticulum, then Golgi apparatus, lysosomes, mitochondria, plastids and vacuoles. All these things we have studied about the cell. Now we come to some other points. One is, we have discussed then the osmosis and diffusion. Osmosis means the spontaneous movement of what? Materials from a highly concentrated region to lower concentrated region, right? Diffusion also in the same way, but the difference is that in osmosis there is semi-permeable membrane. This means the materials are transported through a semi-permeable membrane. Anyway, the movements of materials takes place by these two processes, right? So here we are going to discuss three more points: hypertonic solution, isotonic solution and hypertonic solution. Hypertonic solution means, in a solution, if we keep a plant cell, then from outer region, means from the solution, the materials are transported to the inner region, means inside the cell. Suppose water molecules from this solution moves towards the inner region of the cell, then what may happen? It causes the swelling of the plant cell. Means suppose one example I say, if we keep some raisins, dried grapes, raisins in the solution or in water, normally what may happen? From outer region, grapes, dried grapes or raisins means dried, there is no water content inside. Okay, so inside there is low concentration of water, there is no water inside, there is low concentration. But outside in the solution, the higher concentrated water molecules are there. So from outer region, if we put these raisins in the solution or in water, what may happen? The water molecules may enter into the cell or into the raisins. Alright. So here what happens, these molecules are transported, water molecules are transported from outer region to, or from higher concentrated region to lower concentrated region, means from outer region to the inner region of the cell. So such a movement takes place. Then we can, it causes the swelling of raisins. After some time if we see, then we can see that the raisins are bulged or swollen, alright, filled with water. So here water molecules are entered into the cell. So this kind of solution, suppose if we keep a plant cell in a solution, then the substances are moved from the outer region to inner region, or from a highly concentrated region to a lower concentrated region, then we can call such a solution as hypotonic solution. Clear? Okay. Now hypertonic solution means, suppose if we keep a plant cell in a solution, then some substances are present in the cell. Suppose water molecules are present inside the cell. But in the outer region in the solution, the water content is very low, means lower concentration. Then automatically what happens, from highly concentrated region, these water molecules move out to the outer region where water molecules are lower concentrated. Okay. So then what happens? This plant cell becomes shrink, right? It causes shrinking of the cell. One example, we keep mangoes in salt solution, mangoes, right? Mangoes, in small mangoes we collect and put in salt solution. Then after a few days we can see this mangoes became very or shrinked mangoes we can see, shrinked. Means all the water content from the mangoes released or move out into the outer region. Why? Because in the mangoes or in plant cells, water content is more. But in the sugar solution, water content is very less, because it's a sugar solution, not water. Water content is very low. So what happens, from this plant cell or from this food, the water molecules move to the outer region. So from a highly concentrated region, the water molecules move out to the lower concentrated region. So such a solution, if it happens like that, then such a solution we call as a hypertonic solution. Okay. Now the third one, isotonic solution. Isotonic solution means simply if we keep a plant cell in a solution, but nothing happens, means there is no movement of substances from the outer region to inner region of the cell, or from the inner region of the cell to the outer region, outer or external medium. Then there is no movement. Then such a solution we call as isotonic solution. I think it is clear to you. So then one more point that we have to discuss, plasmolysis. What is called plasmolysis? Plasmolysis means it is the process in which the losing of water takes place. Means the loss of water from this cell. It is called plasmolysis. Plasmolysis, it means that it is the process in which the water loss takes place from the plant cell or from a cell. Suppose from a cell, if water molecules are lost, then it causes the shrinking of water that I told. Such a condition is known as plasmolysis. Okay. these are the points that we have discussed in this chapter. So you have to go through the chapter very well, read very well. Okay, now along with this I will send a note to you. Complete your notes. I mean question answers I will give you day by day. So slowly you have to complete the notes, right? So whatever notes you complete, suppose if I give one note today, then the next today itself you have to complete those. Only few questions I will give and answers also I will give. Just copy these answers. Just copy these answers to your notebook and complete it. And whatever works you got today, you have to complete today itself and send it to me. The next day I will give few more question answers. In this way we will complete all the question and answers of this particular chapter. Okay, I think everyone has understood very well about cell and its structure. Then detailedly you will study about cell in higher classes. Okay, so you will get chance to study very well in structure and all other details of cell organelles in higher classes very well. So anyway, you should study this chapter very well. If you have any doubt related to this chapter, you can ask me at any time, no problem. You can call me and ask. Anyway, complete your questions and answers. You have to draw the diagram of plant cell and animal cell. Okay, plant cell and animal cell, that also you have to draw the diagram of plant cell and label them very well. Two diagrams are there and I will send you the diagrams to you, labeled diagram. You just copy those diagrams. With the pencil you have to draw the diagrams, label the parts very clearly with the capital letters. Don't write simply something for labeling. You should neatly write, neatly draw, and better you draw in an A4 sheet and send it to me. The images you can send it to me. That will be better. Okay, suppose if any correction is needed, then I will tell you. So anyway, complete your work and send it to me. How many days you want? I will give you 3 days. Okay, 3 days time I will give. So till Monday. Monday you can submit all. Okay, so one by one the question answers I will give on each day. And the coming Monday I want to get all the question answers and the diagram. We are going to wind up today. Then do your works properly. Okay, thank you. Have a nice day.